Hello and welcome to this fifth and final lecture in this series on biomolecular modelling. So over the past few lectures we've been looking in quite a bit of detail at the theory and the theory of biomolecular modelling and approaches for sampling potential energy surfaces. Now let's move on to thinking about some practical aspects of simulations, and then we'll be thinking about the analysis and the things we can analyse from simulations. So the first thing you need to do when you're doing a simulation is choose an appropriate force field and computer program. So if you're wanting to study a protein, then you need to choose a force field and some software that can model proteins. The next thing is you need to choose the initial configuration. This is very important because depending on your initial configuration you might get very different results from your simulations. So often what you might want to do is, if you're modelling a protein which has a known crystal structure, you might want to download this from the protein data bank and use your crystal structure as your starting structure. You could also use structures from NMR studies. So NMR spectroscopy can be used to determine structure of proteins. Alternatively, you could use other types of methods to generate an initial configuration, such as bioinformatics, which predict structures based on their sequence using databases. So, assuming you've got your initial configuration, the next thing you need to think about are the boundary conditions in your simulation. Because you can't model an infinitely large system, so how do you deal with modelling just a small system? The most common type of boundary conditions that you will come across are so-called periodic boundary conditions, or PBC. In this type of boundary conditions, you set up a simulation box. It could be a cube, it could be some other shape, and if you had this cube by itself, then there would be vacuum outside the cube, so the molecules towards the edge of the cube would not experience the bulk structure, and they'll be affected by the vacuum. So what you want to do instead is embed this cube in a periodically repeating array of boxes which are all identical. So if you've got a cube, then you can have boxes in all directions adjacent to this box, and it gives you a lattice of boxes. In this type of setup, you will just simulate the central box, but you no longer have edge effects, because when you get to the edge of the box, all it can see is another box. So it's like modelling an infinitely periodic system. When a molecule at the edge of the box sort of moves across the boundary, then what happens is another molecule, an equivalent molecule, moves into the box on the other side. And this means at all times that the number of molecules in the box remains constant. And this is a really good way of modelling a system without edge effects, which is often what we want if we want to study, for example, a protein in water. One potential problem of the periodic boundary condition is if you choose your box to be too small. And if you do that, it means that you sometimes get interactions between the protein in one box with the protein in an adjacent box. If you're wanting to study a protein crystal, then this is fine because you'd have proteins interacting with each other. But if you wanted to study a protein just in water by itself, then you don't want these self-interactions. So this figure here sort of illustrates what's going on. So if we take a central, if we take a molecule of interest here, then we calculate the interactions within this sphere around it. And this sphere is determined by the box size partly. The key thing is we don't want this molecule here to interact with its image here, because otherwise if it can interact with this image and even this one over here, then we're going to get artificial periodicity imposed on the system. And normally we don't want that. Of course, as I mentioned, if you want to model a crystal, then we need this periodicity. But in most purposes, we need to make sure that we avoid self-interactions. So, now we've set up our simulation, set it running, whether we're using Monte Carlo or molecular dynamics, we need to start analysing the simulation to get out various things. So there are various static properties that we can get out, so we can get things like average structures and so on, and they're quite easy to understand what they are. One which is a little bit more subtle is the radial distribution function. This in particular is useful for looking at solvation around molecules, or the distribution of molecules just in a pure solvent. So what we need to do is we take a molecule of interest and we count the number of particles within a certain distance. So in this schematic here, we've got our particle of interest, then we look in a very thin shell between R and R plus dr away from this molecule or this particle and count the number of particles. If we then plot this g of r against distance, if the system were completely uniform, we get this nice smooth curve here, and this smooth curve is just related to the increasing volume of a sphere as you make r larger. This would be if there's no structure in the system. But for most molecules, we have structure. So we'll have a first hydration shell and a second hydration cell, and we can see these in this radial distribution function. So here we have a peak, and then we have a trough, and then another peak. So here we can see that the density of particles around the central particle is higher than would be expected, and then it's lower than would be expected, and then higher again, and then lower, and eventually this curve should just join the bulk line. So this type of plot allows us to look at structure in solutions, so looking at first hydration shells and so on. This way of presenting it isn't actually very useful, so that what we normally do is we divide this function by the volume of a sphere of radius r, and that gives us a much easier thing to work with. So now we have distance across here, and this is our this is our pair distribution function. So this is, for example, looking at the distribution of distances between two oxygen atoms in water, for example, as we increase the distance. So here we have a very strong peak for the first solvation shell, and then we have a trough and then a second solvation shell here, and we could argue that there's a slight trace of a third solvation shell. So this gives us useful information about the structure of a liquid. The nice thing about these radial distribution functions is they can actually be determined experimentally. So you can calculate these pair distribution functions for water, for example, if you do neutron diffraction studies, and this is an area of current research. Then there are lots of time average properties we can look at. So if we're looking at time average things, then we need to be doing molecular dynamics simulations which have a time component. But then we can look at things as a function of time. So one of these is plotting a time series. So this would be just a plot of some property. It might be a bond length, it might be an angle, over time. And we will do this in the practical class. Once we've got time series, we can calculate probability. Well, we can calculate probability distributions, and then we can, using statistical mechanics, calculate free energy profiles from these probability distribution functions. We might want to look at transport properties. So this is things like diffusion constants. So if we were simulating water molecules, we could calculate the diffusion constant of water. And of course this is a measurement which depends on time. We can also measure things called time correlation functions, and these are to do with whether a property of the system at a certain time is correlated to the property at an earlier time. So, for example, if we imagine two water molecules next to each other, at a certain time we would expect them to be aligned such that their dipole moments are pointing in opposite directions, or in the same direction. But as time goes on, we could expect these two molecules to move, and the time correlation function, looking at the correlation between orientations of these dipole moments, could be used to give us information about the dynamics of water molecules. We can also look at things like root mean square fluctuations. So over time, atoms in a system are going to fluctuate. So if you've got a protein structure, we can imagine the atoms are going to fluctuate about some sort of mean position, and these fluctuations are also going to give us some information into potentially function. So there are going to be some parts of the molecule which fluctuate more than others, and it's very easy to calculate these from simulations. So basically, your analysis is going to depend actually on what you want to get out of your simulations. So you've got to decide what's an appropriate thing to analyse for whatever the question is that you're wanting to answer. So that's basically how molecular dynamics, well, how simulations work, whether they're molecular dynamics or Monte Carlo simulations. So what we need to think about now is the problem that affects all types of simulations, and that's the so-called sampling problem. So you can do molecular dynamics for a long period of time, you can do a Monte Carlo simulation for a long time, but sometimes you will just not observe the events that you want to look at. So these are known as rare events, and these are processes which perhaps don't occur in the simulations, and they could be things like large conformational changes. And large conformational changes you probably won't observe in a simulation because it's got quite a large activation energy for the process. But if that's what you want to study, then you want to observe it. So how can you do something in order to observe these processes more frequently? Another example is receptor binding. Most of the time when you've got two molecules jiggling around next to each other, they're not binding, and the binding tends to be quite a slow process, right, which only occurs rarely. So there are a lot of different types of biased simulations or enhanced sampling methods that can be used to enhance the observation of rare events. And all of these methods basically work on the principle that if you steer the simulation in some way, if you put in a potential which pushes it in a certain direction, if you know what that potential is, then you can subtract the effect of that potential later on when you come to do the analysis. So this is the subject of a whole array of different enhanced sampling techniques that you will come across in the literature. The first one to talk about is umbrella sampling. And umbrella sampling basically adds a harmonic constraint to the force field to enhance sampling in unfavourable regions. So imagine you've got a system here, this is your potential energy surface. If you're simulating at a low temperature, then most of the time you would be in this minimum here and you'd be bouncing around here. But if you wanted to get this energy barrier, the activation energy from going from this minimum over the barrier to this minimum, then you need to be able to observe the system up here in order to get appropriate sampling, so that you can get appropriate probability functions, so that you can get free energy barriers. So what we do is we can add a harmonic constraint to hold it at a certain region. So imagine this here is a harmonic constraint which we add to the potential, and that will hold the molecule up here on this high energy region. So it means we can sample a lot better up here. This umbrella potential here would hold us at about this region here, so we'd get better sampling here. And then this harmonic potential would hold us in this region of the curve. So this allows us to do more sampling than we would normally be able to do for high energy parts of the curve, which allows us to get better sampling and then better statistical properties and better thermodynamic properties. So in this plot here we've got 1, 2, 3, 4, 5, 6 harmonic constraints which we add in turn. In reality we'll do a lot more than that, so that we can sample each point on the curve sufficiently to get good thermodynamics, to get good convergence of these values. So as I said, this allows us to produce a more accurate free energy profile. Another simulation approach that you'll come across in the literature are approaches based on what's called parallel tempering. And the idea here is that you run a number of simulations simultaneously at different temperatures. And this is easy because when you have a supercomputer with thousands of processors, you could even run a different simulation at a different temperature on each of these processors. What we do then is randomly we exchange confirmations between these simulations. So in this plot here we've got 5 simulation temperatures. So this is, we're now plotting energy as a function of time, so in a molecular dynamics simulation. And if we plot energy as a function of time for each of these simulations, we get something like this. So I've color coded them so we can follow their process. We simulate for a certain amount of time, and now we want to exchange confirmations. So here we've swapped the blue and the yellow structures. So the structure which was here at the lowest temperature, we now suddenly put into the simulation at T3. And the structure which we did have at T3 is now taken to continue the simulation at temperature 1. What this means is that each of these simulations are actually sampling from slightly different potential energy surfaces. So it means if this is a temperature of interest, then it allows us to, for example, jump over barriers and sample the surface more effectively. Once we've done the swap, the simulation just continues as before, and all the other ones which we didn't swap just continue. So then we do it for a bit longer, and then we can swap other confirmations. So here we've swapped green and yellow, and we've swapped the red and purple. And the middle simulation just continues on. And we can just keep doing this, so swapping confirmations and continuing the simulations. And we just keep going. And ultimately then we can calculate thermodynamic properties for whatever the temperature of interest is, normally the lowest temperature here. So this again just gives us a way of enhancing the sampling of a simulation at a certain temperature by allowing it to explore the potential energy surface more effectively. Another approach you might come across in the literature is our approaches based on so-called metadynamics. So these approaches basically add an energetic penalty to areas of the energy surface that have already been explored. So you can imagine we start off down here, we get good sampling down here, but after a while we've found out everything we need to know about down here. We've got enough data, but we need to know about the other parts of the surface. So what we do is we actually add an energy barrier, or add an energy penalty to being in this region of the potential energy surface. So if in a way this corresponds to sort of filling up this region of the potential energy surface, which will now change the energy surface and push the system over here. So now we sample over here for a bit, and then we can put a barrier in, a penalty, to push it away from that, because we've been there, we've done that. So we can just keep going. So as we sample each area, we can sort of slowly push the simulation away from that regions which have been discovered. So you can imagine we're now sampling up here on the energy landscape, and then we add more penalties, and now we've got to a stage where this basin has been completely filled in. So now when we start simulating, we're going to fall down into this minimum, and we can start sampling in this region of the potential energy surface. So this allows us to sample parts of the energy surface that wouldn't normally be accessible. So you can imagine we keep doing this until this whole basin here fills up and it pushes us over in here. So all of these approaches allow you to get better sampling of the surfaces. And this is one of the major research areas in modern simulations. That more or less brings me to the end of this course. So as always, there's the recommended reading for this lecture, and then I will see you in the classroom.